In 7.3.1, uh, IB would like us just to know that transfer RNA, also known as tRNA, is activated by an enzyme whose job it is to attach the amino acid to the top of this thing called the acceptor stem on transfer RNA. So, and it uses ATP. So we're going to be talking about this in 7.3.1. So let's just review the structure of tRNA. So a couple important parts. So first of all, it's RNA, right? So we have G, 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 C, G, and on the other side, complements C, 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 G, C, right? Here it's weird. We got a U to a G. There's definitely some weird things going on. Sometimes that actually might be a typo. But A to U. But then there's some non-binding sites with some weird molecules that would allow it to have this clover shape. The other thing is like, hey, down here's our codon, right? So the codon is the 3 bases on transfer RNA that will connect to, form complementary bases to the codons on messenger RNA. Right? Codons are the 3 bases on messenger RNA, which is, if you remember, transcribed off of our DNA and carries the genetic code. And these will be complements to this. So if the anticodon is UGC, then the codon is ACG. And this is what we use, not this. This is what we use to determine what amino acid is on the top. So let's talk a little bit about the enzyme real quick. So I'm just going to erase this. So we're going to be focusing on the acceptor stem for this part. So there is an enzyme. So I'm going to draw a little enzyme here. Something like, they're actually pretty big. So let's just pretend this enzyme, we only got a part of it, and it like kind of wraps its way around this whole transfer RNA molecule. But in this little site here, we have an amino acid. So if you remember, we were talking about translation, and the transfer RNA comes in, it drops off its amino acid, and then it leaves amino acid-less, right? The transfer RNA molecule lost its amino acid because it gave it up to form that growing polypeptide, which will eventually be our protein. So it doesn't have one. Oh, it's so sad. So it needs to find another amino acid. So this is what this process is about. Oh, I wish I changed the colors back. So we need this activating enzyme to deliver this amino acid. And it's activated by ATP. So ATP has a little place up here. Actually, I don't know if it's exactly up there, but somewhere on the enzyme that activates our enzyme, which gives it the energy to form a bond off this acceptor stem. They actually want you to remember that the acceptor stem consists of those 3 bases. The acceptor stem, the base sequence is ACC for the acceptor stem. And then the amino acid, it's actually the, let's just draw our amino acid real quick. So C, there's our amine group. Here's our central carbon with a hydrogen. Here's the mystery R group. You guys should all have this memorized. This is double bond to an O. And here we have our OH group. So what happens is the OH group on our acceptor stem off of our base adenine here will interact with the OH group here. This H and this OH will form what? You got it, water, in a condensation reaction, and then we'll form a covalent bond with this O to this the A on our acceptor stem. And that's how we attach amino acids to transfer RNA with the help of ATP.